thank you for the introduction, and we'll let people get situated. So briefly introduce myself. So my name is Joseph Denby. I have a biochemistry background. I've been in the commercial space for just under 10 years now. And I joined CPC Scientific several years ago to help lead candidates progress through the clinical pipeline. And today I'll be presenting on 3 key process innovations for a singular project in a manner which benefited both the client and led to a greener process in tandem. I will give a brief history of CPC alongside and the broader peptide industry. I'm aware there might be people from other modalities here, so we'll do that, and then we'll dig into the actual innovations in detail. So CPC Scientific was founded in 2001 and has taken the standard trajectory of a CDMO that's successful over its lifespan. That includes site expansions, getting a wide variety of processing suites of equipment, and taking the negative positive regulatory steps like obtaining the ISO certification, passing FDA inspections, etc. Then 2016, we started working at commercial scale, and we filed several drug master files accordingly. This has culminated in us being able to take on projects from very early discovery R&D stage all the way through to late stage commercialization, and we have demonstrated that capability over several projects. We have filed over 130 investigational new drug filings, and 13 of those have progressed to commercialization from there. And as you can see here, we have, is there a pointer? You would have pointed in here. Maybe I shouldn't, I'll go without a pointer. You can see we have various manufacturing sites in Hangzhou, China, and a new site in Brooklyn, California, which is set to begin mid-scale GMP production from the beginning of next year. So, as you can imagine, we have projects in the globe, so we have to have the necessary inspections with the local authorities. So we pass inspections with the FDA in the US, the EMA in Europe, the TGA in Australia, etc. And the team is currently comprised of 470 scientists who are all degreed from the different departments, whether that's operation, chemistry, R&D, manufacture, CMC, etc. QC and QA accounts for roughly 25% of our workforce. So, as a bulk service provider, we provide all the activities that are needed from a discovery or clinical lead all the way through to the market. So that the idea is to do that all under one hood, whether it's process development, CMC, guidance, etc. So we support our clients through those stages. As you can see indicated here, we do all the various synthesis strategies: solid phase, liquid phase, fragment process, utilizing standard and canonical amino acids. As a CRDMO in 2026, we have to work on different peptides for different therapeutics, and we have to be able to modify based on the client's needs. So that's from a purity standpoint, but also from a modification standpoint. We also have a relatively newer oligo platform, which we started several years ago, and these are our overall capabilities, very similar to peptides in that we can do quantities from milligram scale all the way to kilo and take things from discovery to commercial. That's headed in Hangzhou by Istas Feng, who developed the China first ASO first-in-class drug, which is in clinical phase 2 currently. So we can do a variety of purities and lengths. We are validated up to 35 mers, but at discovery stage, we've gone up to 80 mers, and we can adjust purity, again, according to client needs. We also offer pretty much any modification that is an IP protected on the market. In addition to solely oligos, we can do a variety of conjugates, as indicated here. So they are single-stranded and double-stranded, and one which I want to highlight is our peptide oligo conjugate work. So, as you can imagine, having 2 integrated platforms is quite ideal for this kind of work, and it does give you CPC some unique advantages in peptide oligo conjugate synthesis. So you can see here how they align. It allows you to do the current manufacture. The synthesis and immediate products can be done in parallel, and that saves weeks off your timelines. So we had the pre-existing peptide platform, which we then integrated essentially. And we can do any of the previously disclosed oligos, as well as the conjugates. We can do modified oligos as well, whether they're pegylated, phosphorylated, fluorescent, etc. So I covered a decent amount of this already, but I'll just give a brief overview of the current state of the peptide industry. So for decades, peptides have always been considered as promising therapeutic agents. They were traditionally considered difficult to formulate due to poor PK profiles in vivo, particularly because they were susceptible to rapid enzymatic hydrolysis. In recent years, advances in rational peptide design and improved delivery strategies, particularly those aimed at enhancing oral stability and intestinal uptake, have helped us overcome some of these limitations and reinvigorated the field. As a result, peptide therapeutics are now one of the most rapidly growing areas in drug discovery. More than 200 peptides are in active clinical development, with over 600 in preclinical stages. And the global peptide therapeutics market is currently projected to reach $295 billion USD in 2033. This rapid expansion reflects both the scientific advancements and the inherent advantages of peptides, including excellent target specificity, high potency, relatively low incidence of side effects, and the ability to treat a wide range of disease indications. More recently, obviously, it goes without saying that the commercial success of GLP-1 receptor agonists like tirzepatide have further accelerated interest and investment. As the demand for peptide therapeutics grows, however, scalability and efficient manufacture become increasingly important. So peptides can be synthesized using several technologies, including recombinant expression systems, SPPS, and liquid phase peptide synthesis. So recombinant technology is highly effective for large-scale production. However, for generic peptide drugs, it can become costly due to extra clinical validation and the challenges of avoiding existing. For this reason, SPPS is becoming increasingly important for industrial peptide manufacture. For example, tetrazetide is synthesized commercially via SPPS. Semaglutide is currently produced recombinantly, but as patent expire and generics emerge, SPPS may play a bigger role in the future. So I'm sure you know, but in SPPS, the peptide is built stepwise on a solid resin through repeated cycles of amino acid coupling and deprotection, followed by the cleavage and the separation and purification. During each stage, extensive washing of the resin is required to remove excess reagents and impurities, and as a result, solvent use contributes significantly to SPPS's environmental footprint. The most commonly used solvent is dimethylformamide, or DMF, which raises environmental and toxicological concerns and is increasingly subject to regulatory scrutiny, especially in the EU. This issue is even more significant for longer peptides. If you take semaglutide as an example, it is 31 amino acids long, so that is 31 cycles of deprotection during the synthesis. So therefore, strategies that reduce solvent consumption, SPPS, could greatly lower both environmental impact and manufacturing costs. Because of this, improving the sustainability and efficiency of peptide manufacturing is becoming a key priority for CDMOs. So for CDMOs that specialize in highly technical fields, every client is going to bring a fresh set of challenges. Across many of the industries in which we work, manufacturing quality and compliance are obviously absolute requirements. That means above all else, we have to meet the highest standards while delivering quality and reliability. They do, however, have that imbalance with the commercial needs of the client, where there's, high purity, but it needs to be under a tight schedule. So even for, say, CDMOs specializing in a specific area, such as peptides, agility and timely implementation of technological advancements are necessary to be able to provide state-of-the-art process improvement. And I feel this is a distinct strength that CPC demonstrates within this case study, where we're going to go into where we successfully delivered multi-kilo quantities of a novel peptide to a client within a short time range, nominally 2 to 3 weeks. And that also delivered in terms of reduced environmental impact and reduced cost, which we passed on to the client. So I'll go through the initiatives one by one. So during this project, like I say, it was a decapeptide, multi-kilo scale, 3-week turnaround time. So we identified SPPS as being the most amenable process for delivery of this material, predominantly because that's our pre-existing expertise. So while we wanted to deliver material to the client, we also wanted to minimize environmental impact and make any savings we could. We therefore looked more closely at the SPPS process to identify ways in which we could make efficiencies. So we identified three key areas. During synthesis of the peptide on the resin, during cleavage from the resin and isolation of crude material, and during purification of the final product. So resin washing is a necessary process during SPPS, and there are 2 points at which extensive washing is required. One is after the completion of the coupling, and one is after the FMOC deprotection. During and after each stage, the resin is washed with solvent to remove any impurities or contaminants, and after FMOC deprotection, it's required to remove residual pyridine. Both are necessary to avoid side reactions that can interfere with the chain elongation process and may mean final purification is difficult. During an SPPS approach, there is significant solvent use. Solvents are estimated to account for 80 to 90% of the overall waste. And of this, the environmental impact of solvents is considerable. For example, on completion of coupling, the resin is washed 3 times, using up to 3 times the resin volume per watt. After FMOC deprotection, the resin is washed between 4 and 8 times, using up to 3 times the resin volume per watt. So if you take something, again, like semaglutide, where you take a 31 for example, assuming 31 deprotection cycles with solvent washes after each step, there's a very large quantity of DMF that's being used throughout that process. So this is environmentally significant and adds substantial financial cost. So anything that can be done to reduce solvent use throughout an SPPS-based process will substantially benefit from a fiscal and from an environmental. So through use of a medal reaction and a comprehensive characterization of waste streams, it was shown that following only 4 washes, pyridine content in the resin dropped from 20% volume by volume to only 0.5%. So in other words, DMF collected from the first wash onwards was 99.5% pure. So it was quickly realized that for this instance, 4 washes would be ample for complete pyridine removal, and that the 4th wash onwards be used as one of the first washes in any subsequent FMOC deprotection. So using this approach, we were able to reduce the total number of washes required after FMOC deprotection, using fresh DMF from 4 to 8 times to 2 to 4 times, and that's effectively cutting the fresh DMF consumption in half. So what did this actually mean? Client. Oh, sorry. So it removed a major operational bottleneck, enabled faster progression, and supported delivery within that time frame, essentially. So we passed those costs on to the client, not through just technical optimization, but a environmental one. So reductive solvent volume required in the cleavage step has received comparatively less attention than other SPPS-based processes, as it is outside that, again, repetitive, iterative process of deprotection coupling. But cleavage is also critically important for delivery of final peptide products for high quality and high yield. The conventional workflow for resin cleavage includes preparation of cleavage solution, which is a mixture of TFA and scavengers, that is then washed over the resin, inducing cleavage. The filtrate is collected and the product precipitated with ease. This is followed by either filtration or centrifugation, then washing and drying to obtain the crude peptide. This process is lengthy and inefficient, placing substantial demands upon equipment and laboratory space and adding significant time and cost to a project. We therefore devised a modified cleavage process that exploited an in-situ neutralization strategy. After cleavage and filtrate collection, the filtrate was treated with saturated sodium bicarbonate solution to adjust pH between 3 and 4, and the solution progressed directly to purification, removing the requirement for isolation of the crude material. Overall, this strategy avoided 4 steps in the previous approach. That's the precipitation in ether, the filtration, the washing, and the drying, significantly reducing solvent usage, saving processing time, lowering cost and environmental impact, as well as improving process efficiency. So again, the impact for the client is similar in that we are removing a bottleneck, so things are moving faster through the process and it's supporting that tight timeline of 2 to 3 weeks. Secondly, by reducing solvent consumption and eliminating multiple handling steps, it lowers waste generation and improves the overall sustainability profile of the process while also enhancing project economic scale. So it wasn't simply a procedural change. Again, it's something that happened to also help with efficiency and stability. So the final stage in the SPPS-mediated synthesis of peptides is the purification and isolation of the crude material, furnishing the desired peptide to the required purity. This is usually achieved through chromatographic purification, where pure product fractions are collected. Often in large volumes that are then subjected to lyophilization, which requires significant time and energy consumption. Improvement in this final step can bring additional cost efficiencies to the overall process. We replace the onerous freeze-drying process with a precipitation method, where product fractions from the separation were collected and concentrated, and the pH adjusted to near the isoelectric point of the peptide. the PI, aka the pH where the peptide is the net charge is 0. This results in precipitation of the peptide, which can be collected using filtration and then dried. So very similarly, this delivered another two benefits to the client. By eliminating that lyophilization, we're removing a significant time bottleneck and allowing faster progression final product delivery. But also, freeze drying is very energy intensive, so we're reducing laboratory resource demand, we're reducing energy costs as well. And an added benefit outside of those parameters is that it also reduces the requirement for specialized lab equipment for that reaction. So the CPC Scientific team delivered several significant savings during this project with positive implication on environmental impact process and substantial cost reductions for the client. Quantities of the solvent used during synthesis were reduced by 36% using a novel DMF recycling approach. Implementation of a neutralization strategy removed 4 steps during the cleavage process. That's precipitation of the ether, the filtration, the washing, and the drying. And a pH adjustment and precipitation strategy replaced the original lyophilization step, accelerating the final purification step whilst maintaining yield and purity. So these combined improvements led to substantial reduction in production costs, and the CPC team delivered kilograms of the desired decapeptide in high purity and excellent yield within a tight time frame of 2 to 3 weeks. So we are publishing a white paper on the initiatives discussed within, as well as some other concepts. I don't know if these QR codes are working yet because we are publishing it in the next couple of days, but I have it. So if it's not working or you want to know more afterwards, please come to booth 75 and I can send you the PDF myself. Lastly, a huge thank you to the teams at CPC Scientific in Hangzhou and California for implementing and innovating these protocols. Thank you to you for listening, and also thank you to Oxford Global for the opportunity to discuss them here.